Hello everyone. In this lecture we will talk about gastrointestinal system development. Firstly we will talk about gastrulation. And the gastrulation, the blast cell migrate through the primitive streak, and the definitive embryonic endoderm cell displays the hypoblast and mesoderm split between the endoderm and ectoderm. Early mesodermal patterning, we have oropharyngeal membrane or buccopharyngeal membrane, and we have caudally the cloacal membrane. A specific region of the epiblast migrate throughout the streak, and different level assume different position within the embryo. We can see here from the midline we have end for notochord, paraxial mesoderm, and then intermediate mesoderm and lateral plate mesoderm, and extra embryonic mesoderm. Developing endoderm is initially open to the yolk sac. The cardiac region initially moves anteriorly, as you can see here, the notochord, the cardiac primordia, and primitive note, chronic villi, and Atlantic diverticulum in the body stalk, and you can see the endoderm and the yolk sac. This huge one is a blood island. Craniocaudal folding at both ends of the embryo, and lateral folding at the side of the embryo bring the endoderm inside and form the gut tube. Again, and craniocaudal folding of the embryo from the oropharyngeal side and caudal part. This will bring the gut forming, and heart will be moved more medially and anterior. As you can see here, folding create anterior and posterior intestinal portals for gut and midgut respectively. Oropharyngeal membrane, you can see the heart, precardial coelom, anterior intestinal portal, you can see, and the posterior intestinal portal. Cardiac region is brought to the ventral side of the developing gut tube. The juxtaposition of ectoderm and endoderm, the oropharyngeal or buccopharyngeal membrane, the future mouth, and cloacal membrane, the future anus. Note that there actually isn't much mesoderm in this membrane, which is important for their breakdown later in development to form the oral and anal orifice. يعني simply ببساطة لأنه الممبرين في الأوروفارنجيال ممبرين والكلواكال ممبرين فيوتشرلي بتكون opening، يعني هيكون فتحة للماوس وللانال canal. For that reason ما في actual membrane in this area. Gut associated organ begin to form but form the endoderm of thyroid, lung, liver, and pancreas. The midgut opening to the yolk sac progressively narrow. By the end of the first month, the stomach bulk is visible. That's very important. Dorsal pancreas has begun to bud. Connection of the midgut to the yolk sac is reduced to the yolk stalk, and then a very thin ventline duct is appear. You can see here from picture A, B, and C. From A, the yolk sac and the folding start with visceral mesoderm, and from B, the connection of the yolk sac to the gut to the yolk sac, and the embryonic cavity appear, and in C, the gut is formed full folded of the endoderm and ectoderm. You can see the dorsal mesentery, visceral mesoderm, and parietal mesoderm. Lateral folding of the embryo complete the gut tube. Mesodermal layer of gut tube is called splanchnic mesoderm, derived from the lateral plate mesoderm, and the somatic mesoderm lines the body cavity. Here, gut tube proper, or derivative of the gut tube. For the foregut, we have the pharynx, esophagus, stomach, proximal duodenum. The derivative of the gut tube from the foregut, we have thyroid, parathyroid gland, tympanic cavity, trachea, bronchi, lungs, liver, gallbladder, and pancreas. For the midgut, we have proximal gut tube proper, we have proximal duodenum to the right half of the transverse colon, and there is no derivative gut tube for the midgut, and for hindgut we have left half of transverse colon to the anus, and the derivative is the urinary bladder. These three regions are defined by their blood supply accordingly. For the foregut we have celiac trunk. We know that from the blood supply of the GIT. For the midgut we have superior mesenteric artery, and for hindgut we have inferior mesenteric artery. The first week to the fifth week, celiac artery supply the foregut, as we said. Superior mesenteric artery supply the midgut, and inferior mesenteric artery supply the hindgut. The figure on the right also shows that the mesenteries, as you can see in the fifth week, the mesenteries and the liver and the stomach have dorsal and ventral mesenteries, where the rest of the gut has only dorsal mesentery. You see between the three arteries, this is the mesenteries has been appeared. Regional patterning of the gut tube. Gut bilayer tube. We have endoderm surrounded by mesoderm. As I said, the gut folding of endoderm with mesoderm. Endoderm is inside and mesoderm from outside. The regional gut tube patterning and organogenesis require bidirectional endodermal mesodermal crosstalk and inductive signals from other nearby structures. Here, the clinicoidal patterning of the gut tube is played out as a regional organ differentiation distinct border form. That means يعني في junctions بيحصل بين الgut tube organs بيحصل فيها change للابيثيليوم during the formation. Okay, like here for esophageal junction or esophageal border, you can see clearly the type of epithelium that covers the esophagus is the squamous epithelium, and for the stomach is the columnar epithelium. Is a distinct border between them. For biologic border, gastric and duodenal junction also the same columnar epithelium but different in the glands. You can see there's a difference. That's the proliferation and differentiation of the endoderm and mesoderm from the different regions of the body. Hedgehog signaling is important for radial or patterning of the entire gut tube. A high hedgehog concentration directly inhibits smooth muscle differentiation by vibration of smooth muscle activating protein, and low hedgehog concentration is permissive of muscle differentiation in the outer wall of the gut. High hedgehog concentration also this is the protein signaling with high bone marrow proliferation which inhibit neuron formation and thus limiting the neurogenesis initially to the outer muscle wall of the gut. Later, development to the hedgehog gene goes away and allowing development of the smooth muscle of the muscularis mucosa and neurons of the submucosal plexus. That means there is an gene like this one is called hedgehog gene play an important role in the muscle differentiation. When there is high hedgehog concentration, that means directly inhibit the muscle differentiation. When there is low of this gene, low hedgehog concentration is permissive of muscle differentiation into the outer wall of the gut. That means high hedgehog concentration is also induced high bone marrow bone morphogenic protein which inhibit the neuron formation and thus limiting the neurogenesis initially in the outer muscle wall of the gut and later allowing the development of the smooth muscle of the muscularis mucosa of the neurons of the submucosal plexus. I hope that's clear. Morphogram induced differentiation of cell fates at different concentration of the first week to the fifth week. Celiac artery supply the foregut, as we said. Superior mesenteric artery supply the midgut, and inferior mesenteric artery supply the hindgut. The figure on the right also shows that the mesenteries, as you can see in the fifth week, the mesenteries and the liver and the stomach have dorsal and ventral mesenteries, where the rest of the gut has only dorsal mesentery. You see between the three arteries, this is the mesenteries has been appeared. Regional organogenesis of the stomach. The stomach appears in the firstly as fusiform dilatation of the foregut endoderm, which undergoes 90 degree rotation, such as that left side move ventrally and the right side move dorsally. Okay, for that reason the vagus nerve follow this rotation, which is how the left vagus nerve become anteriorly and the right vagus nerve become posteriorly. Okay, so the stomach rotate the left side بمشي anteriorly والرايت side of the stomach بمشي معاه posteriorly. إذا الرايت side هيمشي معاه الرايت فيجاس هيكون posteriorly و الليفت سايد بمشي anteriorly عشان كده الليفت فيجاس بيكون anterior. ففي anatomically في الكاديفر when you see a vagus nerve anteriorly to the stomach, that's the left vagus nerve, and that one of the posterior that means the posterior the right vagus nerve. Okay, differential growth established the greater and lesser curvature craniocaudal portion tips the pylorus superiorly. The dorsal and ventral mesenteries of the stomach are retained to become the greater and lesser omentum respectively, and caudal end of the stomach separate from the duodenum by formation of the pyloric sphincter. That's the process of formation of the fusiform stomach. يتكون الميزنتري الدورزال والفنترال ميزنتري واحد فيهم بقى greater and lesser omentum, و junction between the stomach and the duodenum will form the pyloric sphincter, and this is controlled by SOX9 and NXA25 and primary morphogenic protein signaling. Error in this process will not lead to the pyloric stenosis. Pyloric stenosis rather is a common malformation present in 0.05% to 1% of the infants, characterized by very forcible projectile vomiting, non-bilious vomiting in the first hour after feeding, when the pyloric emptying would be occur. Note that the presence of bile would indicate that post-duodenal blockage of some sort. Hypertrophied sphincter can often be palpated as spherical nodule, peristalsis of the sphincteric seen and feel under the skin. Stenosis is due to the overproliferation and hypertrophy of the pyloric sphincter and an error of recolonization, more common in males than in females, so many likely has a genetic basis which is undermined. A closer look to the mesenteries. The stomach and liver are suspended in the mesenteries that attached to the dorsal and ventral body walls. So dorsal mesentery of the stomach become the greater omentum, as we said, and ventral one become dorsal mesentery of the liver and become the lesser omentum. Ventral mesentery of the liver become the falciform ligament. The rest of the GI tract is suspended in a dorsal mesentery of mesoduodenum and mesocolon. Regional organogenesis of the liver and pancreas. The liver and pancreas arise in the fourth week from the foregut endoderm in response to the signaling from the nearby mesoderm. Pancreas actually has ventral and dorsal component, each is specified in different manner. So it's rotated into anti-clockwise from dorsal and ventral pancreatic bud to form the pancreas. Cardiac mesoderm and septum transversum, specific liver and ventral pancreas. All foregut endoderm has potential to develop into either liver or pancreas, but local signals represent the less fit. Mesoderm of the liver, mesoderm endoderm of the pancreas. So this fibrous growth factor bomb morphogenic proteins form the cardiac mesoderm and septum transversum and inhibit signals, but the endoderm is still express as Hoc genes just develop into the liver. Meanwhile, endoderm just caudal to the liver, but in outer of reach from the growth factor and bone morphogenic protein still sees, and so this tissue does not become the liver. Unlike the rest of the endoderm, it does not express this gene and develop into the pancreas. That's the process of development of the pancreas and liver. The dorsal pancreas is specifically signaling from the notochord. Signals from notochord represent S-HOX gene in the caudal foregut endoderm, which is permit dorsal pancreatic differentiation. Rotation of the duodenum bring the ventral and dorsal pancreatic together. Abrasion in this process may result in the annular pancreas, which can constrict the duodenum. Also, since the dorsal and ventral pancreas arise by different mechanism, it is possible that one of the other may be absent in the adult. Rotation of the duodenum also causes it and pancreas to become secondary retroperitoneal organ. Secondary retroperitoneal organ is a structure that was originally in the body coelom, but when got pushed out into the body during the development بيطلع برا. يعني أول بداية كان أثناء الديفلوبمنت كان انترابريتونيوم، ولما خلاص يحصل development قفول للgut وماتوريشن بيحصل له push out. Intraperitoneal versus retroperitoneal and secondary retroperitoneal. We can see intraperitoneal viscera, the yellow one. Retroperitoneal is the red, and the orange is secondary retroperitoneal vessels. Development of the midgut and colon, herniation and rotation. Growth of the GI tract exceeds volume of the abdominal cavity so that the tube herniates throughout the umbilicus. While herniating gut undergoes primary rotation 90 degrees counterclockwise. When looking at the embryo, this corresponding with the rotation of the stomach position of the appendix on the left, and the primary rotation also bring the right vagus into the front, hence the change in the name the anterior vagus. With the growth of the embryo, the abdominal cavity expands and thus drawing the gut tube back within the abdominal cavity and causing an additional secondary rotation. 180 degree counterclockwise position of the appendix to the right. Once the abdominal cavity colon continues to grow in the length, pushing the appendix into the final position into the right quadrant, and attachment of the vitelline duct to the gut at the region of the ileum. Ducts normally represent during development, but not always. Defect associated with the gut herniation and rotation, vitelline duct abnormalities. During the development, the vitelline ducts they can form Meckel diverticulum, or can find vitelline cyst, or can form vitelline fistula. This is the vitelline duct abnormalities of some sort occur in 2% of all live births. Note that this apparent structure are almost always found along the ileal portion of the GI tract. Defect associated with the gut herniation and rotation, we have also omphalocele. This is an omphalocele. Rotation of the duodenum bring the ventral and dorsal pancreatic together. Abrasion in this process may result in the annular pancreas, which can effect associated with the gut herniation and rotation. We can find the valvulus, Meckel diverticulum, fibrous cord, umbilical fistula, valvulus. Fixation of a portion of the gut tube to the body wall. Subsequent rotation causes twisting of the tube, possibly resulting in stenosis and ischemia. This is the valvulus. Development of the hindgut. Derivative of the hindgut, including everything caudal to the distal one side of transverse colon, and distalmost portion sigmoid colon and rectum divide to the cloaca in rectal canal and urogenital canals. You can see here the cloacal membrane and urogenital septum. This is the formation of the hindgut, and cloacal membrane divide into the rectal canal and urinary bladder anteriorly. Errors in the process leading to the imperforate anus. You can see in these pictures A on the imperforate anus, atresia in the B, and fistula in the C, up to F. You can imperforate anus in this A, B, anal is atresia, and from C you can see a perianal fistula, different type of fistula, retrovaginal fistula, retrovesical fistula, retrourethral fistula, and so on. We see the rest of GI tract, enteric neuron and the hindgut arise from the vagal neural crest plus some sacral crest. ده بالنسبة للديفلوبمنت بتاع الباراسمباثيك سبلاي اوف جي اي تي. جزء منه بيجي من الفيجاس نيرف وجزء بيجي من sacral crust. Distalmost portion of the hindgut are further away from and therefore more sensitive to the protrusion and migration. إنه يكون الاريا دي يكون أكتر high sensitive to the migration. Mutation في الجين اسمه RAT ده resulting in the congenital megacolon or Hirschsprung disease. Between the 4 to 6 weeks, the cloacal become the common depository for development of urinary and genital rectal system. The cloaca is quite promptly divided into the anterior and urogenital sinus and posterior intestinal canal by urethral septum. The two lateral fold of the cloacal tissue join the urethral septum to complete separation of the urinary and rectal tract. The effect associated with the gut herniation and rotation, vitelline duct abnormalities. During the development of vitelline ducts, they can form mecal diverticulum, or it can form vitelline cyst, or can form vitelline fistula. This is the vitelline duct abnormalities of some sort occur in 2% of all live births. Note that this apparent structure are almost always found along the ileal portion of the GIT. This is a Hirschsprung disease or congenital megacolon بيحصل في 1 ل1000 ل 500,000 births caused by failure of vagal neural crystal to migrate into the portion of the colon. They innervate regionally and tonic constriction and of the role of the mesenteric plexus is largely inhibitory. Upstream region will become distended, hence megacolon cold. Surgically repaired by remove affected region. That's all about the embryology of GIT tract. Thank you very much.